Hello and welcome. I'm Janet Gillow with Washington University's Brown School, welcoming you to Open Classroom. So happy to have you with us today. I am joined by Cynthia Williams from the Brown School and Amanda Baker from WashU School of Medicine, who will be helping to assist with today's program. For folks joining us on Zoom, welcome. Just reminding everybody you are muted. We can't see or hear you, but we do want to hear from you. Please use the chat feature for your questions and comments as the presentation goes. Also want to note for you that we cannot call on a raised hand, so please type out that full 5. For everybody joining us on YouTube, we are very happy you are with us, and we don't have a direct way of interacting with you. So I'm glad to have you along for the ride though. Before we get started with today's program, I just want to let you know we have another program tomorrow. Dr. Sarah Moreland Russell is going to be presenting on theories of policy change, and that program is offered in partnership with the Brown School's Policy Scholars Program. And then we are taking a break next week, which I'm just a little bit excited about, before rejoining you in April. So I hope you guys also get a break. But now let me cheer up for today's program. This is the cap of our series on the COVID vaccine. And the series has been brought to you by the Clark Fox Policy Institute and the Brown School's Masters of Public Health program. I'm going to be joined later in the program by Associate Dean Rodrigo Reis, who's going to be sharing about a bit about his work related to the pandemic and then helping to lead the Q&A. But our lead speaker today is Dr. David Curiel. He's a tenured professor in the Cancer Biology Division of the Department of Radiation Oncology at Washington University School of Medicine. Dr. Curiel has both an MD and a PhD. And he's been with us here at WashU since 2011. He's the director of the Biologic Therapeutics Center. And his work is focused on gene transfer vectors to advance the human application of gene therapy, virology, and vaccinology. And so depending on the press you read, he's credited as either being the father or maybe the grandfather of the mRNA technology that's behind several of the coronavirus vaccines. He understands the science, technology, and the innovation probably better than anybody else we're going to get a chance to talk to. So here today to share with us the science-driven choices in vaccine design and development, please welcome Dr. David Curiel.
Thanks. I jump in now? Oh, perfect.
Yes, please.
Okay. Pleased to be here. My lecture is going to complement what Janet has put together an open classroom before on COVID. This is the last lecture of the series, and I'm not going to tell you about which vaccine to take, and I'm not going to tell you about side effects, and I'm not going to comment about the variants. What I'm going to discuss is what was the logic that drove the development of the current vaccines, and what is the science that's driving the next generation vaccines. And I'm hoping that understanding the basic science of the vaccines, the biology of how they work, will help you sort through the news and make the developments seem more science-driven. So, first slide. We have to start off with some basic terms as I discuss the vaccinology. And there's not going to be a lot of terms, but there are key terms. The first is the disease, COVID-19. It's caused by a virus called SARS-CoV-2. That's depicted on the left side of the slide. So there's the virus, SARS-CoV-2, and there's COVID-19. That's the disease. The virus has a spike protein, an S protein, and that's the red stalks that you see. And that's the mechanism the virus uses to infect humans. But it's also one of the parts of the virus that our bodies recognize as being foreign and for which we seek to mount an immune response. And such a component of a virus that's foreign, we call an antigen. So there's the virus, SARS-CoV-2. It has an S spike protein that's critical to its infectious capacity. And this S protein is actually an antigen. Our body sees it as foreign. Next slide. And the ways that our body reacts to a foreign antigen is depicted here on this slide. And I'm going to use terms that I know that you've heard in the press when people have evaluated vaccine efficacy. The first phase of the immune response is this foreign antigen is taken up in the body and processed by a cell called an APC, or an antigen presenting cell. That involves processing the protein and presenting it so the immune system can see it. And that process stimulates the two arms of the immune response, phase 2. It triggers the development of B cells, that B lymphocytes that make antibodies. And these antibodies bind and neutralize the SARS-CoV-2. And it stimulates the generation of T lymphocytes. And these are a distinct arm of the immune system. And that generates cells that can directly kill the virus or host cells that are infected by the virus. So our body has two arms of immunity, B cell and T cell, and they defend us against invading viruses by distinct processes. Next slide. So our third term is vaccination. So a vaccine is actually a vehicle, a kind of a delivery vehicle. That's what's used to deliver that antigen to the patient's immune system. And it does it in the way that is the most efficient and effective at stimulate immunity. So as I mentioned, the antigen can be one part of the SARS-CoV-2, like the S protein, or it could be multiple parts. A virus can have multiple antigens. In addition to that, a vaccine vehicle can vary widely. A vaccine vehicle can be a heat-killed version of SARS-CoV-2. It could be a protein portion of SARS-CoV-2, or it could be a gene that encodes a part of SARS-CoV-2, or it can be that gene inside of a genetic delivery vehicle, like a virus. So the logic here is that the vaccine wants to present the antigen to the immune system in the best way. There is a variety of antigens that could potentially be exploited to elicit immunity, and the vaccine vehicle is the agent that presents those antigens to the immune system. No more terminologies. Now we're going to put it together and tell you the story. Next slide. So as I said, the way the vaccine induces protection via immunity is to stimulate the immune system. And it stimulates an immune system that has two arms that can function as defense: the antibody-based B cell or the cell immunity-based T cell. These B cells work by producing these antibodies. I'm showing you that all the way on the left. And those antibodies bind to and neutralize the invading virus. Or the T lymphocytes protect by directly killing the infected cell. So again, the goal is to stimulate immunity, and we have two arms that we could stimulate that can work to protect us. Next slide. The induction of protective immunity is a multiple stage process that can be simply depicted as follows. The antigen is taken up by the antigen presenting cell. The antigen presenting cell processes the antigen, for example, the S protein of SARS-CoV-2, and that processing puts it into a form that the immune system can see it. So here's the logic. The vaccine is designed to exploit the part of the SARS-CoV-2 that's the most potent inducer of immunity. So that we study recovered patients before we've even developed a vaccine, we try to see what their immune system is reacting to. Is the immune system reacting to most potently? And we design our virus to exploit, our vaccine to exploit the best inducer of immunity. So it turns out that for SARS-CoV-2, we believe this S spike protein is the best inducer of immunity. Next slide. So it's important to know that we were designing vaccines while we were studying the disease. The first patients were hitting the ICU at SLU, at WashU, and we were studying them and trying to understand their immune response while at the same time using that information to design vaccines. And the reason is each invading pathogen, be it influenza or SARS-CoV-2 or tuberculosis, has a distinctive pattern of immunity that it induces. So for example, some organisms induce cellular T-cell immunity, and that's the most effective immunity to eradicate them. And some organisms, like SARS-CoV-2, induce antibodies, and therefore that's the best immunity to induce to eradicate them. And some organisms, we want a balanced response where we have both antibody and T-cell. So when the patients come to the ICU and we study them, even in advance of designing our vaccine, we try to understand what is the pattern of immunity of the convalescent patient, and how can we design a vaccine that mimics what our host immune system does naturally. Next slide. So the logic of the vaccine is therefore, and I want to really state this, based on studying patients and understanding the biology of the patients and understanding the biology of the invader. By understanding how SARS-CoV-2 infects, we can determine what's the best antigen. By studying how the host reacts, the convalescent hosts, we can understand what's the pattern of immunity that we wish to induce. And these clinical understandings dictate the logic of the vaccine design. Our goal is to reproduce authentic protective immunity that we see in the host with the vaccine that we design. Next slide. There are a diversity of vaccine technologies that could accomplish this. A vaccine is designed to deliver this SARS-CoV-2 antigen to patients for induction of immunity. But it turns out our immune system is very promiscuous. Those antigen-presenting cells, the APCs, they can take up antigen in many forms. They can see proteins, they can see whole viruses, they can see genes, and this predicates the fact that many kinds of vaccine vehicles could be useful. So this list on the left, this is a partial list of in the works COVID vaccines. This is a partial list. There are more than 140 vaccines being studied. So what this highlights is there's no one best technology, because if we knew that, that would have been the technology that we rolled out. And the evidence is that right now, the lead agents that many of you are getting are either adenovirus or mRNA vaccines. There still isn't full consensus as to which of these are the best. So I want to highlight for you that many vaccines are capable of inducing immunity, and other factors may be the determining factors for what vaccine has the most utility. Next slide. Well, when we talk about vaccine efficacy, there's often a lot of confusion. And so I want to highlight what we look at as vaccinologists. The first thing we do is we use animal model systems to prove that the vaccine works. And the animal model systems have to have certain properties. They must reproduce the biology of the SARS-CoV-2 authentic to humans. So it turns out we can induce mice to do this. We can use Syrian golden hamsters, and we can also use non-human primates. And when we give them the vaccine, we want to show that we induce antibodies to SARS-CoV-2 or T cells, and so we study those. The next thing we do is there are a number of vaccines in human trials. And in these human trials, we also look at the surrogate indices of efficacy. Did we induce antibodies? Did we induce T cells? And finally, because of the reality that for COVID, a number of vaccines show comparable efficacy, they all work sort of well, then other considerations must be considered in terms of endeavoring a full rollout. So again, this is a kind of a scientific feast, if you will, where the initial vaccines all worked rather well in model systems, all worked rather well in human trials, and therefore, as we consider developing them, other factors become more relevant. Next slide. So what might these other factors be? What other capacities of the vaccines do we think about? Well, one is the delivery route. How do we deliver it to the patient? There are multiple ways vaccines are delivered: orally, intramuscularly, intranasally, and each of those delivery routes provoke a distinct immune response. So determining which vaccines work in which delivery contexts and which immune phenomena are stimulated is an important consideration. And I'll come back to this. The other is production. Many of these vaccines are complex biomolecules. They're molecules like viruses that are complex and have to be grown on cell lines. There are molecules that are very, very unstable, like mRNA, and have to be produced under special conditions. There are agents like full virus that is heat killed, and if it isn't fully killed, there could be catastrophic consequences. So production of a vaccine can be a rate limiting step for rollout. We'll come back to that. And then the distribution of that virus is also critical. For some unstable vaccines, the process of cold chain is critical. That's keeping the virus stable and intact from production site to the patient's bedside. And that includes planes, trains, buses to get it there. Next slide. So COVID taught us many, many things, and this learning curve happened fast. And I always like this quote in science, and I use it with my trainees. Julius Caesar said, "Everything of use ultimately derives from war." And the COVID pandemic was in every way a biological war. And so it created many things of use. I'm going to tell you two short stories that show how radically the development of a vaccine against COVID changed the way we do things and understand things. Next slide. So you've heard a lot in the news about mRNA vaccines, the vaccines of Pfizer and Moderna, and how they're now useful and how they're a new miracle drug. But it started more shambolically than that. Back in 1995, I was the first to show that you could use mRNA as a vaccine in a model system. And at that time, there were a lot of problems with upscaling and producing the mRNA, and it was difficult for me to gain industry buy-in. So even though the potential was absolutely clear to a young investigator, it wasn't so clear to industry, and the technology went nowhere. But I did have the positive experience of St. Louis Magazine recognizing me this week. So there's some recompense. So what mRNA embodies as a vaccine is a quick ability to get from knowing the pathogen to a vaccine. With SARS-CoV-2, as soon as we knew the sequence, we could push a button on a machine and it would synthesize a vaccine in several hours. So that's rapid response time. So the urgency of COVID drove full development of mRNA vaccines by Pfizer and Moderna. They solved the problems of production and upscaling. And not only that, the fact that they finally validated it in humans will now drive the use of mRNA for an expanding range of other applications, not just infectious disease, but genetic medicines. So in 1 year, we've gone from a technology that if people knew about it, they'd say it wasn't practical, to now it's revolutionizing medicine. Next slide. So adenovirus is another compressed history. Adenovirus you've heard about as the lead agent for J&J and for AstraZeneca for COVID. So adenovirus had been used for decades to vaccinate mustering in US military recruits. It kept the young men from getting respiratory tract infections in boot camp. However, when the virus was used as a gene therapy vector in 1987, a young man died, and the strong reaction provoked industry opt-out. However, the virus quietly improved. It was used 5 years ago to cure the Ebola outbreak in West Africa. But because of the disease and because of the lingering concerns about the virus, it wasn't developed commercially. The urgency of COVID drove full clinical development. Now COVID vaccines are being put forth by Johnson & Johnson, AstraZeneca, Sputnik, CanSino. So the validation of adenovirus vaccines in the COVID context will now drive the use of adenovirus for an expanding use of other applications. So war has given us new tools, new understandings, new appreciations. Next slide. So our lead agents, so I told you 2 stories of 2 backordered agents that were validated because of COVID and its exigencies. And now Johnson & Johnson, Sputnik, CanSino, AstraZeneca are using adenovirus. Moderna, BioNTech, Moderna, Pfizer are using mRNA. Next slide. So in the lay press, there's a lot of controversy about efficacy and what's the best vaccine. And one of the things I'm not going to speak to at all is what's the best vaccine. I won't comment. I'll give you the tools to answer the question. But as scientists, we speak of protective immunity. We vaccinate a patient and we look at surrogate endpoints. Did we induce antibodies? Did we induce T cells? The next stringency by which we evaluate a vaccine is, did it protect against the illness? If you vaccinated people, did they not end up in the ICU? That's a scientific endpoint. And finally, we look at populations. Did we change lateral transmission? So we look at efficacy in distinct ways. Next slide. So I want to highlight that routes of employment impact the way we understand and use vaccines. So when you give a vaccine in different body sites, it induces a distinct profile. And this profile may have impact in terms of how the vaccine works. We try to recapitulate native immunity. So if the infectious agent like SARS is a respiratory pathogen, it's logical to use a respiratory route for the vaccine. to show how this impacts, next slide, I've collaborated over the last year with Mike Diamond, and we used an adenovirus, and we delivered the vaccine intramuscularly in the same way that Johnson & Johnson and AstraZeneca did, and we got the same kind of protective immunity they got. But when we gave it intranasally, which was a respiratory route, we found that in fact, we got a sterilizing immunity in the upper respiratory tract, and this was superior in terms of protecting the patient. So this highlights that there's a distinct potential in vaccines and the immunity they induce that's linked to the administrative route, and this may have impact for issues like lateral transmission. Next slide. And it also impacts potentially vaccines in development. mRNA vaccines were the first off the block because they embodied this capacity to be made quickly based on genetic information we knew. However, it turns out that nucleic acid vaccines only function well when they're given intramuscularly. So this obligation may limit their potential for other delivery sites. So this goes back to the idea of the best vaccine is determined by many, many factors, not just does it work in an animal model. Next slide. So I want to highlight another facet that goes back to how the COVID situation changed all the rules as we knew them. This is a picture of the NIH where I trained, and it is involved with the FDA in analyzing vaccines and approving them. So normally we'll construct a vaccine and we'll validate it in animal models. We'll go through safety and toxicity testing. We'll do a phase 1 human trial to show the agent is safe. We'll do a phase 2 trial to show it has biological efficacy. A phase 3 trial to show that it is protective efficacy. We'll seek FDA approval. We'll develop it with a commercial partner for production and distribution. This timeline is about 10 to 15 years. There's some people that would tell me that's an optimistic number. Next slide. COVID compressed this into 1 year. And that happened because of aggressive support from the government to leverage key development costs. You usually don't have that. Unprecedented cooperation among commercial competitors. And this compressed the timeline. However, production and distribution issues remain limiting factors impacting full and effective rollout. Next slide. But I want to tell you our story of a compressed timeline. We developed with Mike Diamond a simian or a chimpanzee adenovirus as a COVID vaccine. We got a late start. We started 2 months after everybody else. We validated protective immunity in a murine model. We showed that we had improved immunity by virtue of our intranasal route, and we could use it as a single dose vaccine. We validated this in a non-human primate model. A biotech company in India, Bharat, licensed the technology as well as Precision Virologics here in St. Louis, and human trials are underway in India with our new intranasal approach. And as of this presentation today, we've done 25 patients. So that's 1 year from designing the virus to running this gamut to getting something into humans. And we hope that we will have human data by May that will rationalize fuller development. Next slide. So there still remain choke points with this production pathway. mRNA vaccines require a new technology for upscale and production. Not many places do it. Adenovirus vaccines require an extension of the in-place production capacity, which is currently swamped. The mandate for specialized, what we call GMP production, represents another key choke point. So it's clear that government and commercial investment to expand the infrastructure for production remains a critical point to address in the context of future pandemics. Next slide. In addition, there's the issue of cold chain. This is the point of production to point of delivery pathway whereby a vaccine must remain active and stable. And this requires everything you could imagine: refrigerated trucks, refrigerators in the doctor's office. And the thermostability of some agents like mRNA present much greater cold chain challenges. For example, despite their promise, mRNAs are in fact non-starters for much of the developing world. So clearly, the means to improve cold chain is going to be critical to getting the vaccine to all the countries that need it and to not have a non-democratic distribution. Next slide. So to tie it up in terms of what I think we need in the future, I've told you the story of how we got to vaccines so quickly, and it's a story that's a triumph of science. I've told you the story of all the problems that remain because the needs of the pandemic so swamped existent capacities and infrastructure. And so now I want to look into the future, as assuredly we will have these pandemic challenges again. The mandates for the future are that the biological factors that precipitated COVID remain operative. There will be more pandemics. They will arise in exactly the same way, and they'll be just as potentially catastrophic. The response to COVID established new vaccine platforms, but we still struggled with effective rollout. Even though we could make a vaccine quickly, didn't mean we were going to get it into people's bodies quickly. And this process identified critical choke points that will have to be addressed in the future: production, distribution, cold chain, to make effective the response to the next pandemic. Next slide. So our future design, the futuristic vaccine, will be one that can be designed quickly against new pathogens, one that can be administered by multiple routes, one can be manufactured using in-place infrastructure, and vaccines that are compatible with current cold chain. Just as a for instance, we use Adenovirus, and that's proved to be a useful vaccine, and now we're engineering an unusual Adenovirus variant derived from sheep that embodies a natural thermostability. So future work must address the identified barriers to COVID for effective rollout. Next slide. So I'm going to conclude. I was wanting to give you an overview of the logic of the vaccines we got to and the logic of the vaccines that need to be developed. And I've tried to allow a lot of questions, which I assume there will be. Wanted to thank the people who work with me in the cancer biology division. Want to thank especially Mike Diamond in infectious disease, who did all the animal work. And we had a great deal of help from Baccarat Biotech and Precision Virologics, who remain really vital partners in the rollout of the current vaccine agent that we're developing for intranasal delivery. I'll take your questions now.
So before moving to the Q&A, we're going to have a few comments from Rodrigo and then bring the audience into the room.
Thank you, Janet. David, this was great and very, very educational to all of us. We are very grateful for having you in our community and sharing all that knowledge. Thank you so much. So for those folks that who don't know me yet, so I work on the other side of the spectrum of that. It's not on the vaccine development and technology. I'm working on the sort of prevention mechanisms and sort of the uptake of vaccination and prevention strategies. So doing public health studies. We are doing several studies both in US, but in Latin America. I'm originally from Brazil, which is now the epicenter for the COVID pandemic, as some of you know. And we are learning, we are specifically exploring what are the sort of the systems in place. They are both acting on the spread of the disease, but also on the sort of strategies to prevent the disease to continue to grow, but also on the uptake of the vaccines. So one thing that I would like to say, and just reflecting on David's remark before I give you a few cues of what we have done, people some people are still sort of awed by the fact that we developed the vaccines in just 1 year. And for the scientists, this is not really surprising. So if you look back in history, we did sort of similar remarkable things. Now, if you think that in only 7 or 8 years, we're able to move from one point that we were barely able to launch a rock on the space to put a man on the moon in 7 or 8 years. That was a remarkable achievement for humankind. So when people come together and science is behind, we can achieve things as long as the scientists, they have the support, and they had that at this time. So what we are facing now, and that's one of the things that we learned, is actually science knowledge is not enough to move the needle when it comes to decrease hesitancy for vaccination, and also to help mitigating the spread of the disease. One or two things that we have learned examining countries in Latin America, and they are similar to what is happening in the US, is first, people are aware of the symptoms, are aware of the measures. So educating about what needs to be done is not enough anymore. It seems that part of the reasons that is being for people being more hesitant in either taking the vaccines, but also adopting some of the measures is more political, ideological, and religious based. And that's where we are trying to understand how to communicate to people better what are the possible mechanisms that we can use. That's not only a US solely sort of element, it's happening across the globe. So that's definitely more that should be done there. The other thing that we learned, and we didn't know early on, is that city design and city planning is not really the reason for the spreads. People were skeptical about density in cities, and if you see cities like Hong Kong, well, COVID is not even as bad as you can imagine it has been in the US, where density is not an issue. So city design and public transportation, public transit is not really a major concern for cities. So we are able to operate a lot of the systems we have in place and adapt them for COVID times, and we are adapting them from the post-COVID times as well. So I think we are learning a lot about the strategies to mitigate the disease, but also on how to implement preventive strategies in the future. So those are my few reflections I want to share with you, because I think understanding what's the science behind development of the vaccine increases literacy, but that's not enough. We have to understand how we can actually address the concerns that folks, they are more hesitant in adopting vaccines, and also on adopting strategies to prevent the disease to be spread, to sort of understand them. So Janet, back to you. Should I take some questions on, and what do you recommend me to do?
Absolutely. The audience is already lighting up the chat on fire. So Rodrigo, I don't know if you want to take the lead on some of that, or I can step in.
Yeah, well, let me start with one or two questions here that I think then we can go back to some of there's so many good questions in the chat. So, but an early question we had today, David, people are still sort of want to understand how can we be sure that we have immunity coming from a vaccine? how we actually can tell that this is really working or not.
You're saying once you get vaccinated, how?
Yeah, how you can prove the vaccines provide immunity to get infected.
Yeah, when you do your animal studies, you validate that the vaccine induces a given kind of an immune response, and you even study the dynamics of that response. You can study how long the antibodies last, how long the T cells last. So the presumption that the agent you get is active and that you will respond is an implicit assumption we all make. I guess if you were really worried, you could look at getting a test of the antibodies to COVID, and they should rise in a predictable way, and your readout should be a clear index that you had a response to the vaccine.
Well, I think that's a very good segue for the second question, and to bring sort of a follow-up. So for how long is the immunity no good for? So do we need to require another vaccination rounds next year? When we need to get new shots?
No, it's a fascinating question, and obviously a really practical one. And this is another one of those questions that we're in the process of answering and we haven't answered yet. But what we've already learned, for instance, in the phase 2 trials, in the context of declining antibodies, that the antibodies that were induced by the vaccine, the protective antibodies, had risen, plateaued, and were on the decline. Those people were still having less infections than the people at plateau. So we know that the level of the antibody isn't completely predictive, because it may be that that T cell or cellular immunity plays an important role later in infection. So the unsatisfying answer is we don't exactly know how long the vaccines last. They probably last longer than the intervals we're given them for. And I think that the field is looking for annual vaccination. And probably the tools that we have in hand are already capable of that, but haven't been fully validated in that regard.
That only reinforces the need to suppress the disease now and get people vaccinated as fast as we can, I guess.
Yeah, obviously people invoke influenza, where we can know enough about the disease annually to design a vaccine annually that we as a population all receive. So that would be a conclusion consistent with how vaccination is done in this country.
So I have a few more questions that I want to ask you, David, from the chat. But if you allow me to address one question that was raised about Brazil, if you don't mind.
I want to take that question.
Yeah. So I think we can do that, work on that together, though. Somebody's asking here about what should we doing to work with Brazil to make sure that we really have coronavirus under control. They are mostly working with Sinovac. Well, actually, they are working with AstraZeneca as well and Sinovac. Well, and they just signed contracts with Pfizer and other providers, but I'm not quite sure the question is about the coordination between the US and Brazil. I think the one thing that the opposition in Brazil is claiming the government to do is to work with US with the extra shots that are reserved here and to be bought by Brazil to be delivered right away. Because the issue in Brazil is not delivery capacity, it's an issue of inventory. There's no vaccines available there. So the system is different set, and David, you know that better than I do probably. So the vaccination, immunization process in Brazil is quite effective, but right now there's no inventory available. Am I right, David?
Yes, I agree. And I think it's also important to note that the vaccines that appear the best right now may not end up being the ones that have the greatest utility. As the father of mRNA vaccines, I should have more of an emotional commitment. But I'd be the first to say that the cold chain issues are so overarching with mRNA that something needs to change to make them more stable, or else they're simply not practical for most of the world. And that will be a consideration in terms of what ends up being the best vaccine. So I don't think we've seen the best vaccine yet, and I think a lot of work will go into it. I think one of the stories I really hope the audience got was that the two vaccines that ended up being lead agents before COVID, those were Bush League vaccines. Those were second tier. Nobody was using those. So it was surprising that those ended up being the most effective vaccines. And it took some creativity in terms of the scientific community to revisit what was seen as an older technology.
I think you just answered Richard's follow-up question about what does he was implying in terms of Brazil and variants and the available options we have for vaccines. So thank you, Richard and David. Brilliant as usual. So may I just bring up to your attention two things that are sort of relate to each other. One, you mentioned earlier in the talk that a death was related to the initial adenovirus, vaccine. And somebody was asking, what has changed in the adenovirus, technology that is making that technology safer now? And so any comments?
A really wonderful question. And I will respond to Janet's invocation to tell personal stories. So I've worked on adenovirus for 30 years, and I'm an expert in this virus. And when this young man died in 1987, there were people who said my career was over, that nobody was going to fund this virus. But I was fortunate enough to be funded. The NIH supported the work, but it was a matter of the scientific community couldn't see the improvements that happened, because scientists have preconceived notions too. And I always, the young man's name was Jesse Gesslinger, and I always told the story that no matter what I presented on, no matter where I presented, somebody would always ask me about this poor young man who died in 1987. I had to keep explaining that. And now I say, after COVID, you know what? Nobody has brought up Jesse Gesslinger, because the virtues of adenovirus were so clear. But there was a long period where the scientific community wasn't open to this virus or its virtues because of one event that was reacted to idiosyncratically. So I'm sure there's other prejudices in the field, and there'll be other technologies that out of this kind of war setting reveal their virtues.
I think you're just making the points. The good science is always transparent, accountable, and very open. It takes me to a second question that I want to bring to your attention. So, there's sort of a skeptical view among several minority groups across the globe. Here in the US, particularly among the Black population and people of color, because of the history of post-World War II, history of, mismanagement and unethical practices, as we all are aware of. So what has been the participation of minorities in the trials? And can we assure that the vaccine is effective across the board for all ethnic and racial groups?
Well, you know that we respond biologically different to vaccines, men and women, and different racial groups. So knowing how we all respond and how each population responds is an important scientific question that we want to answer. But then there is also the whole issue of vaccine hesitancy based on these unfortunate historical things. And the ways that this has been approached by working with leaders within those communities would seem to be the best means. But it's a problem because of some of these historical wrongs. And if the whole population isn't vaccinated, then we're all at risk. So it's a problem for the people who have hesitancy, and it's for the problem for the people that are living amongst them.
That's wonderful, David. I think now as a scientist, I think we all believe that transparency and clearly addressing the issues they're facing, those challenges, is the best way for us to overcome any challenge, right? So I think... And
I think what you were alluding to, I think the political leadership is so critical when it's a population problem. And again, Janet said, tell personal stories. So Merck built a football-sized facility, football field facility to make adenovirus for vaccines in the 80s. And then when Jesse Gesslinger died, this unfortunate death, the consequences were so strongly experienced that the company said, we're not going to make this anymore. And they had an empty football-sized production facility. And it took President Biden saying to them, now you're going to make J&J's vaccine because you have the capacity to do it. So I don't think Merck on their own would have said, hey, we've got this unused facility. But once political leadership spoke to the better angels of their nature, we expanded what was a choke point, which was the production capacity.
Yeah, I think that's a wonderful point. And definitely, again, leadership, transparency, and being very, addressing the issues, not avoiding the issues of deflecting. I think it's important. There are a few questions here regarding sort of effects of vaccines, and so I'll just bring two of them to see what's your reaction. One is why people have different reactions to different vaccines. So why the side effects seem to be slightly different. That's one of the questions on the chat.
The response to a biological agent like an infectious disease is linked to your genes and your response. Mm-hmm. So it's logical that your response to a vaccine would be as well. And we're finding more and more that the side effects that are observed often have biological explanations, and often those biological explanations come down to genetic differences. Mm-hmm. So just like we're using genomics powerfully to determine who gets disease and how they respond to therapy, we'll be able to use that same technology that we're leaders in here at WashU to start gauging why people respond to vaccines the way they do.
Yeah, I think that's a good point. People also should remember that it's just like, if you think about any, cold and flu, we all have different reactions. So we don't react the same way always. That's in the individual response to any agent, right? So there's somebody's really, asking about blood type and vaccines. What's this interaction? What's your take on that?
Blood type, I admonished the audience that I wasn't going to get into these kinds of specifics. But I know that blood type is an index of a genetic composition. So it's a marker for a genetic background. And there is data that different blood types have different reactions to the vaccine. And I can't comment specifically, but I'll say that the informatics services for COVID are wonderful and unprecedented. The Mayo Clinic, Johns Hopkins, the CDC, the NIH, all have fabulous websites for these kind of detailed questions.
Wonderful. Questions are coming just like popcorn on a hot pan, so it's great. So I have another one here. It's quite interesting too. So Bonnie's wondering about what's the scientific merit of somebody taking a mRNA vaccine and an adenovirus one.
There's a fabulous scientific merit, and we call that the booster effect. So when you give somebody a vaccine a second time, they have a better response. It's called an amnestic response. But you don't want the response to the vaccine vehicle to confound that. Mm-hmm. So the most beautiful picture is they'll see the antigen with vaccine 1, and they'll see the antigen with vaccine 2. So that's a beautiful use of the technology. And we're pursuing that with Buckbrot for the South Africa variants. And other companies are now looking at exactly the strategy that the questioner mentioned, which is boosters, adenovirus plus mRNA. And of course, the Russians did that off the bat. The Sputnik vaccine was 2 different serotypes of adenovirus that were genetically distinct. So there's lots of logic to that.
Yeah, wonderful. Those questions are really, really good. I have another great question here, probably the last one we have. So Violet is now wondering, what would you tell to somebody that is concerned about how fast vaccines were developed? I think you mentioned that early in your talk, but maybe if somebody's asking, how can I trust somebody was so, something was developed so fast? What's your take on that?
Rodrigo, I think you answered that beautifully, is that scientists always knew we could do it this fast. And it was the downstream development costs and regulatory processes. And the government responded. So we've developed vaccine. We're always in a hurry. We always want to be the first to show something, and we can make vaccines fast. What we don't always get is the resources quickly. So in this instance, for example, the NIH established a rapid funding mechanism, and we were lucky to have our work funded quickly. And then they established, they made their infrastructure available to us. So normally to do a primate study, I'd have to write a grant, I'd wait months, and the NIH called us and they said, we read your paper, send us the virus, we'll test it in our primates. So it was an example of the downstream processes working as well as they could. And I think everybody has learned something. We can do this fast again if we need to.
Well, obviously, I agree 100% with you. So we are sort of reaching the end. So any closing remarks you want to share with us?
I think that people should access information sources like the Brown School has set up. This was a fabulous series. It covered so many facets of the pandemic. I watched all of them, and I think it answers a lot of the things that people are worried about. I think there are other good information sources, and I've mentioned them. The websites, CDC, NIH, Hopkins, and you can go there. And I'd say that social media is the worst place to go to get information when there's so many good and legitimate sources. And then I'd say that even though it was horrible, science came through triumphantly, and it can again. And we're going to create infrastructure, and we're going to create mechanisms, and next time we'll do a lot better. You know,
I share your optimistic take on that. And scientists, if we're not optimistic people, we probably would never do that, because your career is a testimony of how important it is to have a vision and believe in the future, and things take time. So my own reflection.
Or what you said, and to get people to buy into your vision. But yes, I think it's true. Scientists working together was the characteristic thing that made this go so well.
Yeah, wonderful. No, we are almost ending. David, it has been wonderful. Before getting this back to you, Janet, I appreciate all the questions and your patience. And it has been a remarkable time in history. If you look back 12, 13 months, it's hard to believe that we are reaching the point now. 12 months ago, we couldn't believe that we would have a vaccine. Now we are talking about how to convince people to take one, because we don't have an inventory problem anymore. We are reaching the full capacity of delivery. So I think it's a wonderful problem to have, and I'm sure we will be able to do that as a society. We have done and overcame challenging times before, and we will be definitely doing this again. Janet, on to you now.
Well, thank you so much. I can't help but in listening to this feel just a tremendous debt of gratitude to the scientists who had vision before we even needed it and were prepared to work, to the public health professionals that were implementing strategy, to those early patients whose volunteerism helped pave the way that a year in, we are in a brighter place. To our audience, oh my goodness, so many smart questions, not enough time. Thank you all for being a part of this. And to that end, thank you also to my co-hosts, because I don't think we've ever hustled quite so hard to bring as many thoughts into the room as we did today. A question had come into the chat about watching the earlier programs in this series. All previous Open Classrooms are both on our YouTube channel and on the Open Classroom webpage at the Brown School. If you click on the tab for past classes, you can catch up with what you missed. But with that, we are at time. Thank you all for spending this hour with us. And we hope to see you again very soon at another open classroom. Please stay healthy and safe out there. Goodbye, everybody.